Good morning, everybody. Team 11, thank you for joining us. So we're basically going to start doing these meetings, I think, once per month. The Zoom monthly meeting, weekly meetings we were doing before. I think right now everybody's kind of busy putting together between your current obligations plus putting together all these INDs. It's taking a lot of time. So we don't want to waste anybody's time, and we'd rather concentrate on the pending INDs. I think I'll turn it over to, so as you know, so we're right now, I think we're up to 14 or so INDs, two of which the plasma is already ongoing, and then the BCG is in that period of 30-day waiting to start. And then we have a lot of others in progress that we expect to submit in the next few weeks. So with that, I'll turn it over to Saurabh, and then Saurabh, you can start introducing the different team members that have a report today. I think you're muted, Saurabh.
Okay, so we have quite a few INDs going on, and we've been working to get the BCG going, and we are currently in the process of writing up the IRB. We have it about 95% complete. We have also worked to finalize a consent letter for it. We have a translation in Spanish, thanks to Jennifer, and the IRB itself, we've had a lot of help on that with Taarak and Harsh and Jorge. So the IND for BCG, it discusses a clinical trial. It's a single-blinded study. It's on prophylactic BCG vaccination. We're looking at inducing immunomodulating effects on the innate immune system, specifically in healthcare workers. So we will be enrolling up to 500 local healthcare workers at the Larkin Community Medical Center. And we will then move on to another BCG vaccination trial that will look at aerosolized recombinant BCG. So we have principal investigators on board for both of these trials. For the first one, we're going to be working with Carlo and with Pedram, who are chief residents in IM at Larkin. And then we also have Alina Alvarez for the recombinant BCG vaccination study. And then Carlo and Pedram are also going to be working on this recombination vaccination study. After that, we will be working on a biostatistics project. So we're looking to represent prognostic modeling trends of COVID outcomes in patients in the community and also in the uninfected populace to see the rates and be able to educate the public based on the prognostic rates that we design. We're going to make it a data-based approach. So we have quite a few people who can help us with that, like Dr. Rapolu, Dr. Kaul, and Karun, and Meghna will also be working on this. So Meghna is also working on the zinc silver solution project, and she's going to be leading that. And the zinc silver solution project discusses a partnership with a company called D Orbital, and they have expressed interest in a project at Larkin where we combine a solution of zinc and silver cations and assess the efficacy of prophylaxis and prevention of SARS-CoV-2. So the PI for this project will be Pedram. And then after that, we will have quercetin, which is a protease inhibitor. It'll be compared with saquinavir and with atazanavir. And that'll be discussed today by Jorge and Jordan. Then after that, we also have a tocilizumab review going on. It's an interleukin-6 inhibitor, and we've almost finalized it. That'll be covered by Jennifer today.
Let's go back a little bit to the. Oh, you're gonna have them talk about each of them subsequently, or?
Yeah, definitely.
Okay, good.
Everyone will present. I was just giving a quick.
The overview. Okay, got it. Okay.
A quick overview. So we also have a trial going on for a look into UVBI. So UVBI we will specifically be focusing on the UVA and UVC range. That's being led by Alex, and we're currently finalizing about a PI. Then we have NAC as well, N-acetylcysteine, which will be compared with IV glutathione, and that's all being co-led by Preet, and we have a PI named Talia who's expressed interest in working on that project. We have a almost finalized dual IND for plasmapheresis, which is basically plasma exchange therapy, and it can also be autologous exchange therapy. So we're specifically focusing on plasma exchange, and we will effectively remove the plasma that contains the infected virion particles and replenish it with fresh frozen plasma. We'll do that in one trial, and then we have another trial comparing that to Medrol. So we have PEX and Medrol, and Karun and Karthik will be explaining that today. We have a PI on board for that, whose name is Tashi. Then we also have a trial looking at the efficacy of hydrogen peroxide as a mouthwash for prophylaxis against SARS-CoV-2. And Ala is taking charge of that. Then we have a new IND idea today presented by Dr. Patel. We also have another IND idea on pathogen recovery therapy presented by Dr. Sajwani. And we also have a new review paper idea by Dr. Akano. So we can start today by talking about the biostatistics project. Dr. Rapolu, Karan, and Magnar here. It would be, yeah, you can take a lead on this.
So we're waiting for who's going to do the biostatistics project?
Meghna or Karthik or Dr. Rapolu are here. Where is Karthik?
I see Karthik.
Hi, I'm here. I'm Meghna.
Hi, Meghna.
Was Dr. Apollo not able to make it to the meeting?
Yeah,
Let's see.
I'm audible.
Oh, he's right here, Dr. Rapolu, right here. Yeah. Hi, Dr. Rapolu. How are you?
Hi. I'm fine.
Okay, I'll introduce him. So Dr. Apollo is a computer scientist, and he's working with the University of Hyderabad in India right now. And so he was brought on board by Saurabh and another doctor called Dr. Rajan Patil from SRM University. And so he already worked on a model for prediction of COVID infection and recovery rates, and I think death rates as well. I'm not too clear on that. He's gonna talk. And, can we hear from you, Doctor? Sorry, he's not a computer scientist. Can we have you talk about that more, sir?
Yes.
Yeah. Okay.
So we started this model. Am I audible, right?
Yes, we can hear you.
Yeah. We have started this model about past 6 weeks, and we were able to forecast infected and death rates for the upcoming week. We have tested it for the whole India country in a narrow level. Like we have done it for states and districts. The districts are like if you can think it as counties there. So with a decent accuracy, we were able to give a generic model for India. We have also tried for Italy and Russia, like few cities there. They're also the forecast for good. So in the case of Miami-Dade, we tried, but there is a data lack. Like we were not able to find the recovered data. So in the new normal dashboards also, the discharge data is there, but it was available from only 6th May. But the epidemic started around March. So we need the past data also to give more better accurate results.
Doctor,
So
can you give me, or maybe work with Megna? Megna, maybe you can give me the list of exactly what data you need, and then I will request it.
Yeah, sure. I took a note of everything Rapolu or Taarak said, and I can forward that to you, Dr. Mitchell.
Yeah, I can definitely seek that data.
Okay, all right, great. That's perfect.
yeah, once the data is there, we could immediately give that results, like whether it's currently working or not in that case. And we would also like to know the other modeling members in this group so that we could collaborate for like to increase the pace in this case in the Miami-Dade modeling. And the Texas model is there, right? We have gone through that also. It's based on the social distancing measures taken from an app. I confirmed with Meghna that there is no such app in Miami-Dade for now. So I think we have to go with the database model that we have. And if the app is available, we could later incorporate in it. For now, we only need the recovered data. In the new normal dashboards, it's available from May 6. We need the past data as well. Once that is done, we could give the results whether they are working or not.
That's great. So, yeah, so as soon as I get that data, I'll request it. The request for the data, I'll request it. Perfect.
Yeah, sure.
Okay, so next we will have the plasmapheresis project, and Karthik and Karun will describe that in more detail. Basically, we have two INDs, and they're both almost complete, finalized, and we're looking to compare plasmapheresis as a mechanism of therapy in severe COVID patients, and also mild and moderate. And then we're also going to compare plasmapheresis as the exchange therapy with fresh frozen plasma to the effect of IV Medrol, sodium succinate. So they will explain it.
Thank you, Saurabh. So as Saurabh said, we have two INDs for plasmapheresis, and IND7 will compare the effect of plasmapheresis versus combined Medrol and plasmapheresis. Both the IND and IND8 will be, simply comparing plasmapheresis with the standard of care therapy. And Karthik will be talking more about IND8, so I'll focus on IND7. IND7 is almost complete. Now we're just trying to do the final editing and also get on board with a PI. So we'll be dividing the patients in 6 different groups. Each of them will be focusing on moderately ill patients, severely ill patients, and severely ill patient with ARDS. And each of those 3 groups will be further subdivided to make a total of 6 groups. One of these groups will have standard of care and plasma exchange therapy, while the other will have standard of care and plasma therapy, plasma exchange plus Medrol, solely Medrol actually. So yeah, that's pretty much it. I mean, we'll be trying to see the effectiveness of this therapy to basically remove the cytokines as well as virus from the blood of the patient and see how it works out. So yeah, that's pretty much it for IND7. So if Karthik could tell something about IND8.
Yeah. Hello, Dr. Mitchell, and hello, everyone. So this is Karthik. So for IND8, we have completed all the sections and just finalizing on the reviews to be done. So basically what Kevin has told, we are doing is the same. We are dividing the patients into 6 groups. So they are like moderately ill, severely ill, and severely ill with ARDS. And each group is further subdivided into 2 subgroups, where one subgroup is given standard of care with the plasma exchange therapy, and the other group is only given the standard of care. so that they can be used at the center. And so since we have completed all the sections and just the reviewing is left, next I'm planning to connect with internal medicine resident at Larkin so that they can be helping me joining as principal investigator. And I think from next week we can start working on the trial. So that's it from me. Work with that.
Perfect. So after that, we will be working on UVBI, and Alex is going to be leading that. And is Alex with us right now?
Yeah, I'm here.
Okay, perfect.
So basically, right now, the person who we're using the device from is working to figure out what needs to be done to get an IND from the FDA. He told me he was going to try to get back to me today on what he figured out with that. The main real holdup for the whole process is just getting the IND done, and we have most of the IRB that's needed already done. And that's about it.
Okay. Yeah, that's fantastic. So we are also doing the tocilizumab review paper. It's the interleukin-6 inhibitor. And Jennifer has been leading that.
Hi, hello. So yeah, we are working right now in the tocilizumab systematic review, but we're also looking about a possibility to work in a retrospective multi-case report that we were talking with Dr. Michael and Dr. Marcus. So right now, the tocilizumab review have 11 possible studies that we can work with. And we're trying to see, actually I sent it an email to Dr. Marcos 2 days ago to see if that's enough to work with it. Also, we have been, we had saw that in UK, University of Nottingham and University of Johns Hopkins are making another systematic review. That will take a year. So Dr. Mikel, I don't know if that will interfere with ours because that's a kind of duplicity, you know?
Jennifer, repeat that again. I'm sorry.
Yeah, we were like looking around, you know, there's a site that is, the name is Prospero, in which you have to register your systematic review. So in that site, there's already one systematic review registered about tocilizumab. It's in UK, and with the sponsorship of University of Nottingham and Johns Hopkins University.
So you're saying that could interfere with ours?
Yeah, it's a question. Actually,
No,
because
I'm sorry, because what?
we have, right now we have 11 studies, but you know, there's a lot of clinical trial going on. So they will do their systematic review in 1 year. Ours can be smaller, but it will be sooner.
I think the main thing is if maybe run it by someone that knows statistics and that can at least give us some idea of how valuable these observations are. You know what I'm saying?
Yeah.
Because basically it's going to depend on the journal that you submit it to. So I've seen stuff get submitted with, you know, to journals, maybe not the bigger journals, but some of the smaller journals, and they could get published. I think at the end of the day, we have what we have, right? So we don't have all the data that they're going to have, obviously, but we could. There's no reason why we can't use our data and try to make something of it. I think the main thing is maybe run it by someone that can tell us whether someone who's published before this type of review paper, to see if this is something that a journal will pick up and publish. I think that's the only question.
I already asked it to Dr. Marcos. I sent an email to him. I thought he was going to be in this meeting, so it will be easier to ask him. But then I'm going to wait for his recommendation. Also about the
Sorry, is there anyone in the group that's done review papers before?
Yeah,
Like,
definitely. So I've done review papers, and actually we're working with Dr. Gonzalez right now on a convalescent plasma paper, and most of the group is working on that. That's being led by Umar and Uzam, and also Krunal and Karthik just finished up the conclusion section for that. Dr. Sajwani is also on that paper. She's going to present today about pathogen recovery therapy.
I have Dr. Sana. She's the pharmacologist. She has a specialty in pharmacology and also have a background in in research. And she told me that it is not the quantity of actual papers, but the quantity of patients they have inside the studies. So if we work with that, we can follow up with the systematic review because we will have like 200 patients sort of. So yeah, that's the idea. We're working in that.
In my case, I wouldn't wait for anybody. Just go ahead and put it together and submit it.
Okay, perfect. Yeah. Also about the multi-center case report, right now we only have the information of the South campus. We have 9 patients total, but 2 of them, the information was lost somewhere. So we don't have information about it, so we have 7. And we are waiting for to have remote access in the data of the Palm Spring campus that they have 29 patients with tocilizumab right now.
Jennifer, send me an email requesting that, and then I will connect you with the IT personnel.
Yeah, actually, we already sent the formula to Orlando. And he actually answered back saying that he was going to do that, but that was 2 weeks ago. I think he has a lot of things in his plate because he's accepting the email. He's doing the emails for the contact tracing group, so maybe that's why.
No problem. Just send me a reminder so that I can move it along.
Perfect. Thank you.
Thank you.
So, yeah, that's tocilizumab.
So after that, we are working on the quercetin trial, and this is a natural protease inhibitor. We're comparing it with saquinavir and atazanavir. And Jorge and Jordan will tell you more about this.
So good afternoon, everyone. Good afternoon, Dr. Michel. The quercetin trial, IND, is almost, I want to say 95% or above complete. There are some questions that we had that we wanted to address with Dr. Michel specifically regarding some aspects of the protocol and inclusion criteria for upper age limits in this population. Jordan also has some other questions that we want to discuss with you today. Is he in the meeting?
Yeah, I'm here. So I guess we just take it one at a time. So the first question was, we know that coronavirus is affecting most lethally the elderly population, and the older you get, the worse it is. And so for that reason, we were thinking that in this study, we should be including all ages above 18. However, it'll also change our morbidity, mortality, and as well as the amount of other conditions that we're going to be dealing with in our patients. So Dr. Michel, we wanted your input on whether or not you think we should have an upper age limit, if we should set a limit at 65 years or not.
Why don't you just break them into cohorts, like monitor outcomes at different age groups, because that might also be more telling.
That's a good idea. Thank you, Dr. Michel.
Yeah, so break it down by age group so that you can say, okay, for people that were under 60, people that were from 65 or 60 to whatever the number is, 75, and then above that and above 85, and just see, because maybe that'll give you a different indication, good or bad, who knows? But I think it's a good parameter to so that we don't lump all the data in, right?
Right.
Do you agree with that, Saurabh? Do you think that's a good plan?
Yeah, definitely. I like that. Maybe we'll do a nested case control study if possible. That way we can account for any ongoing patients, or maybe we could look specifically at those who are getting infected ongoing.
Okay.
So after that, anything else about quercetin? Jordan, Jorge?
Yeah. Okay, so regarding exclusion, no, sorry, we already resolved that. We'd like to proceed in contacting Roche, Myers Squibbs, and Corsigen Pharmaceuticals. And one of the things that's holding us back is understanding how funding works, since George and I are new to this, relatively new to this. So when we're on the phone with them, do we need to immediately address funding costs, things like that?
You're on the phone with whom?
We're not on the phone with them yet, but we're planning on reaching out to Roche, Myers Squibbs, and Corsigen Pharmaceuticals regarding their products.
Yeah, I mean, at the very least, we would want them to at least cover the cost of the medication. If they, some will offer to cover some other costs, but I mean, I think at least if we get the cost of the medication, I mean, some of the medications are very cheap anyway, but the ones that are expensive, depending on what the cost is, like I know tocilizumab was an issue until we became part of that trial, because it cost several $1,000s. But yeah, other than that, I wouldn't give them too many requests, because a lot of them are not. They're not emphasizing this particular use, or this may not be. Generally, when they fund stuff, they fund it based on what their priorities are as a company. Like they do this strategic planning. So when we come up and say, "Oh, we want to see if this works or that works," they're not gonna probably have funds for that. So at the very least, we just request that they provide the medication. You know what I'm saying? But that would be probably enough if they do that. Does that answer the question?
Yeah, I think so. So when you say provide the medication, that's with them billing us for the medication, or providing it covers all costs covered?
Generally, they'll just provide the medication free of charge when it's part of a trial. But obviously, depending on the cost, like if some of this stuff is very inexpensive, like the quercetin, for example, so we wouldn't mind covering that. I wouldn't be concerned about that one. Like anything that obviously costs as little as quercetin, probably that wouldn't be a big consideration. But something like Sofluza, for example, even though we've been using it, it starts adding up when you use a lot of it, especially if you do 500 people or 1,000 people.
Okay, thank you. And then I think this is the last group of questions that we had. We needed to know for our protocol, what determines a positive patient? Is it one positive nasal swab PCR? Is like what is the Larkin standard for what evidence do we need to consider a patient positive?
But including that, not only positive, but what do we say negative is also?
Okay, so positive is a positive PCR test, which is basically a nasal swab or oral swab, not antibody testing, but a positive PCR test. That's positive. And then I guess negative would be a negative PCR test. We can't mix it with antibody testing and all that, because it just becomes more complicated. Does that clear it up?
Yes.
That's clear, yeah. And then for resolution of disease, what's the standard to determine resolution of the disease?
You can go by a variety of measures. Some people just monitor symptomatology. Other people say PCR negativity. So it could be either of those or all of those factors.
Okay, so there's some variability at Larkin in determining the resolution of disease. Okay.
Yeah. For example, there's some like, for example, the trial with plasma. The standard is, and it's not even our standards, it's kind of using plasma in the first place. It's 14 days after no symptoms and a negative PCR test, or 28 days after no symptoms. No PCR test required. So as you can see, so there's a lot of variability in how that's determined. The problem with the PCR test, honestly, is that sometimes some people can be negative after 2 weeks, and other people can be persistently positive, a month later. We don't know why. We don't know if that means anything. So in a way, repeat PCR test is not necessarily, a very reliable criteria to determine whether somebody recovered. You see what I'm saying? Because some people are perfectly fine a month later, haven't had the symptom for a month, and maybe all they had was a headache and a fever a month ago. And they're perfectly fine, but they keep testing positive. So I wouldn't say that they didn't resolve their symptoms just because their PCR keeps testing positive. I mean, the disease, because I think a more reliable estimate of disease is actually symptomatology, because this variable with the amount of time it takes to become negative on PCR, and it might be just some strands of virus that are left that may not be active. You see what I'm saying? So
Yeah, I
that's
understand.
Yes, I do. I understand. Thank you. That
No
clears
problem.
that up for us. I appreciate that.
No
Thank
problem.
you, Dr.
And then the last point that we wanted to address prior to moving on to the next IND, our team discussed tracking outpatient subjects every 7 days. So doing blood tests at day 1, day 7, day 14, day 21, and then going on to day 30, 45, and 60. Is that a practical way to keep track of outpatient subjects, or will that be too much? Is that too little?
It seems like a lot. And the only reason is just to bring these people back in, especially when they're feeling fine. I would try to spread it out a little bit more. It sounds like you have them every 7 days, right?
Yes,
Sorry,
every
repeat that
week
last
right
part
now.
that you said.
It sounds like you're having them like almost every 7 days, right? Pretty much.
For the first 3 weeks, yeah, every 7 days for the first 3 weeks.
Yeah, are you basing that on another trial, or are you basing that just on what you guys thought as a group?
That was just what we came up with as a group.
Yeah, I just think it's a little too frequent, and we might have a problem with compliance, getting them to actually show up for the test, because you're asking them to come in for a test, right? So
Yeah, exactly.
yeah. So I don't know. Maybe every 2 weeks might be a little bit more reasonable, for them to actually come. But I don't. It's hard to say. Maybe try it every week and see what happens. I mean, if not, we'll just use the data every 2 weeks, right? I mean, the worst that can happen is
Right.
that they may not show up every week. They might show up every other week. But there's no way to know for sure. So I guess, yeah.
All right, we'll take that into consideration when we're putting that together. Thank you for your input, Dr. Michelle.
No problem. Thank you.
Thank you, Dr. Michelle.
Thank you.
And unless George has anything else he'd like to present, then I think we're good to move on.
No, I think you covered all the questions pretty well, and we got the answers that we were looking for. So thank you.
Thank you, George.
So next, afterwards, we are working on the zinc silver solution, and that will be led by Meghna. So she's here and she'll explain.
Okay, thank you, Saurabh, for the introduction. So we almost are ready with the IND. So I think everyone's already contributed sections and they've uploaded it onto the doc. We have a meeting scheduled for today at 7 p.m. where we're doing a group editing session. Dr. Michelle, I know you wanted it by Sunday. We'll be ready to submit it to you for primary review tomorrow. So you said you wanted to reach out to the T orbital company, right? So I think
they've been pretty they've been calling me every day. So yeah, that's fine. So tomorrow is perfect.
I'm really sorry. We'll definitely have a draft for you to review tomorrow. So if it's ready to submit it to them after you take a look at it, then yeah, that would be great. Apart from that, I think Jorge has already gone through the draft we have already, like right now. He's gone through it once and he's made a couple of comments, and most of them have been resolved. And Jennifer also, I think, has gone through the draft once, and she's already also made a couple of comments. And I think the section members need to resolve them as of yet, but we'll definitely be ready by today for sure. Also, one question I had was, throughout the protocol synopsis that D Orbital had provided us, they keep talking about a baseline, and they say that in the exclusion criteria, that if the patient, so their inclusion criteria says that the patient would have had to have tested or confirmed positive for COVID-19 by the PCR. But then in their exclusion criteria, they would mention that patient is tested negative for COVID-19 within 96 hours of baseline. So are they saying that these are patients who initially tested positive and then on a second test within 96 hours, they're testing negative now? That was kind of confusing.
Who's the question for?
Anybody that wants to answer it. No?
Hi, sorry. Sorry, can you repeat your question again? Hi, by the way, everyone. Good afternoon.
Oh, good afternoon. Thank you. So basically, we had a protocol synopsis that was provided by the company that initially reached out to us with the zinc silver solution. And they say that in the inclusion criteria, it's a patient should have tested positive for COVID by a positive PCR test. But then their exclusion criteria mentions that the patient should have, an exclusion criteria would be a patient who tested negative for COVID within 96 hours baseline. And I'm assuming baseline, that that's their initial PCR test.
And, I believe there is a nasopharyngeal PCR, like what Dr. Michelle mentioned. But keeping in mind, there is also false negative results when it comes to nasal test compared to sputum. There was a study done, I was looking online, and it mentions that when it comes to false negative results compared to nasal secretion, pharyngeal, and sputum PCR, they found that sputum PCR had low false negative results, like up to 11%. And I think when we are looking for a recovery, a person is disease-free, if that's the criteria, if you want to look, it has to have a low false negative result. I think the best, I honestly don't know, how 96 hours from the baseline, if it's negative. If they use sputum, if there are any cough present, if sputum is productive, in those patients, you can actually check and you can assure there is a low false negative result. That generally shows disease-free population. I hope that answers your
Magna,
question.
can you repeat the initial part? Because what I understood was that you're confused whether there's an initial positive in the inclusion in the inclusion criteria is a PCR, a positive PCR, and then exclusion criteria is a negative PCR.
Yes. So I think what I'm trying to say is they mentioned baseline. So okay. I'm confused about what they mean by baseline. Does that mean a positive PCR at admission?
Yeah, I think that they're referring to when the patient is providing informed consent. After they've provided informed consent, when you do the initial test, if they test positive in a PCR, then they would qualify for inclusion into the study. But if they test negative within the past 96 hours, I believe was what you said.
Within 96 hours from baseline.
From baseline, right? So let's say that you. Oh, but I don't. Yeah, that's confusing because then that would. Yeah, are they saying that you're going to repeat? Are they saying that you're going to repeat the test 96 hours after? Or within 96 hours?
Yeah, that was my question.
If it's stated that it's after, if it's at baseline and then 96 hours after, then they're insinuating for you to repeat the PCR. But if it's within 96 hours of baseline, I would take that as within the past 96 hours before they provided informed consent, which would disqualify them because, that 96 hours is what, 4 days more or less. That would mean that they've tested negative 4 days prior, I think.
Okay.
Hey, Megna.
It's confusing. You're right, Megna. You might want to just ask them directly. Do you have their email address?
I think it should be there somewhere on the synopsis.
Why don't you just ask them, because I'm confused by the term baseline.
Yeah, I'll just ask them. I'll send them an email and see if they respond.
Yeah, copy me just in case. Okay, Sanjay, go ahead.
Okay.
What I made out of that was that the PCR should be positive within 4 days. So for like example, for some drugs we have taken 72 hours, right? So what they mean might be that instead of 72 hours, they made it 4 days. Instead of 3 days, they made it 4 days. So if they do not test positive within 4 days of screening, they are not included in the study. So that's what I made out of it.
Maybe. Yeah, that could be.
Okay. So
But
I'm just if it's before so is this 96 hours before is baseline after 96 hours or before 96 hours? Okay, let me rephrase that.
No.
I'm going to ask.
Be like,
sorry, yeah.
No, what I meant was that it's like from the screening time, the baseline from the screening time. So it's when the patient actually presents for the first time.
Okay.
So that's usually like we take 72 hours for like different drugs. I think for zinc, they mentioned 96 hours instead of 3 days, maybe 4 days. I think that's what they meant by it.
Okay, so should I ask them whether from the point of screening, are they looking at 96 hours behind? So like, okay, so are you trying to say that when they first present at our hospital, like I think who was it that mentioned the signing the confidentiality document?
Yeah, that was me, the informed consent.
Oh, right, Jorge, thank you. So I think that what I'm gonna ask them is, are we looking at the 96 hours prior to signing the informed consent that they need to get a PCR testing positive, or is it 96 hours after that, right?
Mm-hmm.
Yeah, because I would think that in order to meet the criteria for inclusion in the study, they would have to have a positive test in COVID, no?
Right. Okay.
Yeah.
Yeah, I'll just let you guys know. Thanks. Apart from that, I think that's all we have for now. Yeah, we should, we'll be ready by tomorrow to submit a completed draft, yeah.
Hi, Dr. Michelle. I wanted to update you for the convalescent plasma review paper that we've been working on. So we're almost compiled up the draft, and I'll be sending to Dr. Marcos as he suggested that he'll review it, and then let's hear an opinion from it, and then we're all set for publication for any journal also.
Thank you, Mark.
So that paper will be sent as a review, and it'll be regarding early convalescent plasma usage. So after that paper, we have another possible paper coming out, and that'll be, it's a new idea by Dr. Akano.
Thank you very much, everyone. I am going to be as brief as possible with this. So it's basically hinged on a statement, a release statement, which was on the 9th of June this month by Dr. Maria Van Kekkel, who's the World Health Organization technical lead team leader on COVID-19. She made a statement during a Q&A session asserting a fact that symptomatic patients are not infectious. And I consider this to be a very huge statement. Dr. David Samadi referred to this as a 360-degree turn. I mean, considering the effects which this could potentially have in terms of easing of lockdowns, social distancing, rules being degraded, so to speak, and the downstream effects it's had on economies of countries, job security, schools, and other forms of academia. And so Dr. David lays claim to certain articles from Singapore published ones, which I mean asserts this fact that asymptomatic patients do not necessarily or are not infectious, and so the need for a lockdown or social distancing really isn't present. So a few days after, Harvard Medical School issued a release where they claimed they had evidence suggesting people without symptoms can readily spread the SARS-CoV-2 viral particle, which is the agents causing the COVID-19. So this really places the world in a state of dilemma. Where do we actually stand? Do we stick to the statements by the World Health Organization team leader, or which points do we exactly stand? So I see this as a big question which we need to answer. And for this reason, I was hoping we could attempt a literature review on asymptomatic patients and the risk of transmission of the virus, or we could perhaps initiate a cohort study. It wouldn't actually end at this, and it might not necessarily have to just be restricted to a literature review, as there are other possibilities which we could employ. Or I just thought we could, a literature review shouldn't be a bad start. So it's just an idea which I feel we could pursue, which is we wouldn't possibly be the only ones, trying to seek answers in this regard. Several other, papers I believe would currently be worked upon as we speak. So I just felt we could perhaps pursue this as a potential research question, which is, the risk of transmission of COVID, of the SARS-CoV-2 virus in asymptomatic patients. So thank you very much.
So you're proposing to start a new, basically this would be like a review paper, right?
Yes, sir.
Okay, because there's some people in the chat asking to join. Do you have anyone? Actually, first, Saurabh, do you have any suggestions for him?
So I think we could do a country-specific model, because the transmission risk in asymptomatic patients will depend on the country's policy. Like in New Zealand, currently they have 0 active cases. Vietnam, they actually have less cases than in West Virginia. So it really depends on the actual social distancing policies enacted by the government. And it'll be difficult to make a general, broad, non-specific transmission risk model. So I believe we should do it specific to each country.
What are your thoughts on that?
Basically, I very much agree with all he said. Just that we could create a simplistic model where we assess perhaps the risk of transmission for any 2 individuals in close proximity, what we refer to as close contact, that's within 6 feet for over up to 15 minutes. So if perhaps we could equally create a simplistic model, irrespective of the country's policy, it still would apply to every single, potential hypothetical case of transmission.
I think the detail is going to be important. Like how do we, right now we have a contact tracing thing going on. We could just use data from that, right? So we could identify of the people that were contact tracing, how many, what percentage were exposed to people who were asymptomatic, Saurabh, something in that. I'm just trying to figure out how we're going to do this. First of all, where we get the data from. And I think the only reliable data we have is the people that we're testing. So we could do 2 things. One is for everybody that we contact trace, what percentage of them were exposed by asymptomatic people? Is that does that make sense? Or
Yes,
Yeah,
sir, it
I think
does.
that's feasible. Definitely a viable idea. We would just need to have data of how many people were in contact with the people who were contacted by the tracer, because I think that'll be one important factor. Like, what is the range of social connection that the people we're in touch with actually have? So I believe that would be an important variable in determining transmission risk. If we can get that kind of data, I think it's feasible. Since this is almost a statistical paper, I think Dr. Rapolu would, I would want to hear what his take is on this too.
Dr. Rapolu?
Yeah, I'm there. Could you please repeat it again? The exact pronunciation.
Sorry, can you repeat the question?
So we're thinking of looking at transmission risk in asymptomatic patients proposed by Dr. Akano, and we were thinking of using the data acquired from the contact tracer team, and this team effectively assesses who people that have been infected with COVID have been in touch with, and that their range of social interactions, we could possibly assess that and thereby judge the transmission risk. So what do you think? Is this doable, feasible? Dr. Rapolu?
Yes, it is. Yeah, it is feasible, but I don't think it will give. It is still a rough estimate, I feel. Like,
Okay.
we don't get a real one.
Okay. That's fine.
And one more thing from the model part, like, what are the expected deliverables from this model?
Did you say expected derivatives?
Deliverables.
Yeah.
So Dr. Rapolu, so with the modeling, what we're seeing obviously in Florida is that there's been an increase in the number of cases, and some of that has had to do with, people having the perception that it's okay. And so we don't, like, if you look at what the government puts out, it's all very comforting about how things are better, and even they even dismiss the fact that so many more people are becoming positive. They're saying it's because more testing is being done. So I think the purpose of the model is if we can have a reliable model where we can tell people, if you do these things, right, such as social distancing or different parameters, this is the percentage of possibility that there will be X number of deaths. I think that would be very positive as far as changing people's behaviors. So that's what we're hoping to do with the modeling. It's kind of obviously, we want to make sure that it stands up to, because people will look at it critically, particularly government people can look at it and say, oh yeah, this doesn't make any sense. But we want to make sure that whatever we put out there is easily indicted or questioned and reviewed, but that eventually it becomes a model that they can say, wow, this model is really predictive. Look, whatever they're predicting is happening, and therefore people should heed the warning, whether it's right now we're not seeing a lot of social distancing in Miami, and it's mainly as a result of what the government is saying about it, the curve being done and no more issues. Does that clarify, Dr. Rapolu?
Yes, sir. Yes.
Yes, sir.
And one more point is that is there any reports like the ratio of positive to the hospitalized cases? Any rough estimate on that in Miami?
Other than what we're getting from that link I sent you, that's the only data that's out there. We could request more. I could talk to the mayor's office and we could ask for whatever data you would like. I could say, we would need this data in order to give you more actionable information. But right now, all we have is what they're posting in that website.
Yes, sir. I could see that the positive cases are like the hospitalized is comparatively low than the positive. Like the people are less admitted. The rate of admission is less compared to the positive cases. Like
Correct.
I feel there is a lag in the positive cases and when they get admitted to the hospital.
Correct.
Like I would like to know the intervention strategy there and also the quarantine statistics. Like how it goes. In case of India, we know, but in the Miami, exactly how do the how do they treat the patient after once he gets detected positive?
So right now, what's happening is that there is no real contact tracing going on. The state says that they're doing it, but we don't think it's happening. And we think that's one of the issues. But that's kind of one of the lessons learned that we're hoping will come out of this, is that if more of that is done, we can show what would happen to the both the positivity rate and also the mortality or the hospitalization rate.
I have something to say. So I was talking to Taarak in the morning, and I think what he's trying to say is basically in India, everyone that tests positive is being, I think they're being hospitalized. So it was easier for them to project that in their model. So he was confused as to such a big discrepancy in the number of positive cases and the hospitalized patients in the US and like Miami-Dade County as well. So he was actually asking for, I think he was asking for the ratio of the people who actually tested positive and the hospitalized patients, because he said that would help him do a projection model much better, like more accurate.
Okay, I mean, we know how many people are diagnosed positive and we know how many people get hospitalized. And we know obviously that not everyone that is diagnosed positive is hospitalized. So we could get those numbers.
Yeah.
So the next topic is the BCG3, the recombinant vaccination. And Dr. Patel has been working very diligently on that, and he would like to present.
Hi, everyone. All right. So so far in update of IND3, again, I'm just going to give a brief review, brief title, name. It will be a single-blinded, randomized, controlled phase 1B, 2A interventional clinical trial. And we are assessing safety, immunogenicity, tolerability, and efficacy of aerosolized recombinant BCG in healthy individuals. So there are certain challenges that I also would like to discuss, after speaking to Dr. Alina, who is an assigned PI for this project. Now we have so far 3 recombinant BCG strain that we found, but only one is from local here, and the headquarters is in Baltimore. The thing is, contacting manufacturing company or biotech company has been a big challenge. I spoke to Serum Institute of India in Pune just to propose an idea if we are changing the formulations of intradermal VPM 1002, which is a recombinant BCG phase 3 clinical trial they are running in India, in Pune particularly. And I spoke to the person who is handling manufacturing, and they want a detailed proposal of content, like what we are expecting. But after speaking to Saurabh and speaking with looking online, challenges will FDA allow this, because when it comes to trading law and phase 1 and 2 combined trial, will they approve this even if they say yes, recombinant BCG here in the United States? So that's a number one challenge. Second, recombinant BCG strain is called ARAS. It's also a good, which is combined with perfluoroalkyl O and BCG, and it has a better bioavailability and better immune function when it comes to preventing respiratory pathogens. I tried to contact them, but I couldn't. They say the line is not under existence. So I'm planning to find as possible way to get in contact with them. So we are in a dilemma that we have a content ready, because the majority of volunteers have worked pretty well in putting in content, but we are still have to find out which will be a better option. Where the strain which is here available in the United States has no any safe clinical trial done on humans. Phase 1 was being conducted in Missouri, but it discontinued for some reason. Compared to international recombinant BCG, there are clinical trials all running in second and third phase. So here, that is the number one challenge of finding manufacturer. Second challenge that I wanted to also mention, that as we know, this is a new route, aerosolized recombinant BCG. We have to do, and it is controlled, interventional group. So I'll tell you the study design. Interventional group will have, there are three different subgroups. We are going to do a dose escalation study of these aerosolized recombinant BCG, where we'll be given low, medium, and high dose of this new formed vaccine, which is already there in different another form, intradermal. So, and control group will be aerosolized saline, intradermal non-recombinant BCG, which is BCG ties with pharmaceutical companies here. Merck is a manufacturer for that. And there is a recombinant BCG intradermal. Whatever we are going to propose, a formulation in an interventional group, another control group of the intradermal will be there. So 3 control and 1 interventional with subgroup with a dose escalation study. Population will be 18 to, we decide actually 21, because according to American Pediatric Association, up till 21 age is considered a pediatric. So 21 to 60, we have decided the targeting populations. Study duration will be 6 months. Now there are certain time frame of what are the outcomes will be. In primary outcome is looking if this new aerosol recombinant BCG has a better immune response by checking quantification of BCG, checking cytokine stainings, and even humoral response, because here we are targeting mucosal antibody IgA, which is also present in plasma. So that is, it has a better response in animal studies. So primary outcome will be safety, which we are going to monitor adverse event up to 1 month. It'll be self-reporting, and patient will be anywhere going to be followed up for certain other secondary outcome we are going to check, like looking for quantification of BCG. So we need a bronchoalveolar lavage for aerosolized patients. So it might require invasive procedure like a bronchoscopy and intubation. And people who have generally received intradermal will require a punch skin biopsy, where we are going to quantify the amount of BCG and the local innate immune response. So it is a very good novel approach. One already study that I found in Germany, they're already conducting in South Africa. They already have aerosolized recombinant BCG, but it is a viral vector and BCG antigen on that. But that's, again, it's an international vaccine. So 2 invasive procedures, skin biopsy and bronchoscopy will be done at certain time frame. It will be 0 day, 14th day, because we believe that BCG, after introducing BCG, that strain stays intracellularly at least up to 2 weeks to create immunogenic response. So 0, 14th, we decide then 28, 90, and 180 days. So this will be 5 times, there'll be invasive procedure will be done during the 6 months of extent of period of the study trial. So total number of people who will be required bronchoscopy will be 25, because we are keeping 5 people in a subgroup for dose escalation in interventional. So that becomes 15 people in interventional. They will require bronchoscopy for just a bronchoalveolar lavage, so we can do check secondary outcomes. And total 30, I believe 20 people, but 10 people will be in control group, each control group. So 30 people in control, 15 in interventional. Now in control, 2 group will be, we are going to check punch biopsy. So 20 people will get punch biopsy. So total throughout the study, if we look at 100, punch biopsy will be done, and bronchoscopy will be 125 times. Now it is when it comes to a cost concern, I wanted to discuss with you, Dr. Michel, if content is ready, if you, with your permissions, if cost and plan looks appropriate, we can just further go into details.
So we're talking about costs of bronchoscopy?
Overall procedure will be 125 total, 125 times of bronchoscopy throughout the 6 months. 25 people means 15 people from interventional group, and 10 people will be from aerosolized saline that we are going to give from the control group will require bronchoscopy. 20 people means 10 people in BCG ties, intradermal, and 10 people will be recombinant BCG, whatever we are going to use as an interventional. Their form of the recombinant BCG will also use in skin to see which one is better. So 25 people requires bronchoscopy and 20 people require skin biopsy in this procedure, and 5 times, 5 follow-ups will be there. So it will be 125. There'll be anesthesias, and bronchoscopy will be involved. Even in skin punch biopsy will be just a local anesthetics, and you can take a biopsy of the superficial skin. It won't be that invasive compared to bronchoscopy.
Yeah, what I would do in that case, Manu, is I would send me that so that I can run it by our pulmonary director, the director of our fellowship in pulmonary diseases, because I want to make sure that he is okay with that before we move that further, because he'll be the one that will be doing the procedure. So I want to make sure that he's
so one study in UK has already been done, but it is a non-recombinant BCG, just a simple BCG, and knowing at the confounders, place and prevalence and incidence, it looks like they were doing bi-weekly. They did for 3 months bi-weekly bronchoscopy and biopsy, on patients. But knowing that this is a new formula and new formula, it could be the nebulized or it could be just a mist. That's something a biotech company, we need their opinion as well. So cost, total number of procedures we have discussed, we have made even a very tight protocol as much as we can when it comes to inclusion and exclusion criteria, keeping in mind that it is a phase 1 clinical trial. And actually phase 1 and 2 both combined. We are checking 4 outcomes: efficacy, first safety, immunogenicity, tolerability, and efficacy of this new drug. Right now there are VPM 1002 is a very well-known strain, and there are 6,000 healthcare workers are going into phase 3 clinical trial. So the chances of it conferred the safety compared to other conventional BCG, but it's in India. I don't know if FDA will allow, even after they will not have a problem creating this formulation, because it's a biotech company and it's a largest production in Asia. They are producing, Serum Institute of India. So I spoke to him. He wants detail, but meanwhile I'm also contacting FDA. I spoke to FDA today morning, and they want us to submit a request of informations just to seek that if they can, because this is not for a treatment purpose, it's just for a clinical trial, if they will allow any international strain or not. So we are working on that too.
That's going to be important. Yep.
So far that's the update. Otherwise content is pretty much done. We have put right now 2 strain, VPM and ARAS, both in a content. Whichever strain will find, will get a green light, or once we get a hope, we'll delete the other content. It will be ready to submit after we hear from them. but I will send you the revised FDA IND, 3 on your email. And yes, it will be wonderful if it can be reviewed by pulmonologists, just to see, because as we are, introducing this new interventional drug in lungs and through aerosol route.
Okay, perfect.
Thank you, Dr. Misha.
Thank you.
Oh, sorry. Another concern that Dr. Alina Alvarez also mentioned, like we are doing BCG trial, but as we know that in Miami, 80% of populations are immigrant, Cubans, and if they are in inclusion criteria, we are involving BCG naive patient. So that means whoever has never received BCG, so it will be a very low fraction of people who wants, first of all, the invasive procedure, and finding the target population will be also challenging. But we are only including 45 people, and that not geriatrics and pediatrics, only healthy adults. So what are your views on, she asked me to, cross-check with you, and what challenges, will be there in the Miami areas, especially, around Larkin communities.
Around recruitment of,
yes.
I mean, I think, yeah, the bronchoscopy part, I mean, if it's skin biopsy, I don't think it's going to be a big deal, but I think bronchoscopy, just convincing them might be. That's why I want to kind of talk to the pulmonary doctor, because once he, if he gives us a green light and he feels that, he supports the study, then I think it'll be easier for him to sell it to people. But I mean, our community is pretty similar to any other community. I mean, as long as the doctor is saying that this is a possible option and that we're studying the disease, I don't think we would have too much of a big deal. We just have to get the doctors, recruited first.
Yes, I agree. Thank you.
No problem.
All right, so the final topic is pathogen recovery therapy, and it's a new proposal by Dr. Sejwani, and after that will conclude the meeting. Dr. Sejwani? Okay, I believe she's on the participants list, but if she's not here, we can talk about it next time.
Yeah, maybe she's muted. Actually, I think I see her here.
Yeah.
Maybe she can't hear us, or I'm looking in the chat. So while we wait for her, so with the new, what are we doing with the new members? We're having a lot of people coming in and they're asking if they can be on a group. Can we come up with some simplified way to redirect them as far as?
Definitely. Krunal has been working on that, and he will be able to answer this the best because he's been working more on the group organization. So, Krunal, are you ready? Hello, Krunal?
Krunal, can you?
Yeah. Hello.
Yeah, so just explain a little bit about how we're going to manage the new member intake and the group organization and where we can direct it.
Okay, so what we can do is the group links are being sent to Dr. Michelle already, and when these people, the people who are interested in joining the teams, you can just send them the links and they can get added to the groups. And whenever we'll have a new group or some new project, we'll put it on the main group.
Perfect. So for new members that are listening, and anybody that's gonna hopefully later listen to this meeting, just so you know, we will be posting those opportunities. So if the groups are full, you can always sign up for the new ones.
Exactly. So we need people to join the main group, and then there we Saurabh or someone can keep posting the new ideas or projects so that people can and once they are on the group, we'll assign them tasks. So that would be much more secure, and everyone will get an opportunity to do something.
Excellent. And so maybe we'll, unless she's ready now, do you want to try one more time, Saurabh?
I just contacted her. I think she's ready.
Oh, go ahead.
Dr. Sejwani?
Maybe she's still muted.
I told her to unmute. I'm not sure. Okay. I think
Meanwhile, meanwhile, I have a new, I know we have a lot of volunteers, in a group. There is a one new study that I wanted to propose to, here in this meeting. Because as we know, pandemic is not over yet, and there'll be many, more cases that will be seen. On some days, you will see like a rapid rise in entries of patients, diagnoses and hospitalizations. So it's a new study where we decrease the hospitalization rate. First, I wanted to, do a study to work on the people who are already admitted. It's about a natural strategy to, I don't know, until we, wait. So is the right time to discuss with, or we are waiting for Dr. Sejwani?
Go ahead, Manu.
Okay. So it is about the astaxanthins. So recently, I remember in last meeting we discussed about the pigment, that it is a type of xanthal flavonoid that you it's a carotenoid actually we found in marine source, and there is a good data has shown that it works better when it comes to reactive oxygen species or any kind of free radical induced damage, whether it's any kind of oxidative stress, could be infection. Is the number. When it comes to COVID-19, we are going to focus on that. There recently a company we call Cardax has taken a patent about they are checking astaxanthin and coronavirus disease of 2019, specifically working people who are admitted. So they already taken patent where they're going to compare with the corticosteroid and astaxanthin high dose, just to see if which one is work better when it comes to acute disseminations, when patients, before they go to ARDS, all those biomarkers like CRP, interleukins, and everything they're going to assess. So the main, they have taken patent for astaxanthin's effects in coronavirus disease. So they are going to include people who are already diagnosed with COVID-19. Now, I looked back in yesterday, and there are certain studies shows that astaxanthin is a powerful antioxidant. The thing is, so far, any lipid bimembranes, when we actually make it thicker, that can actually interfere with the viral entry. Not only that, on any double bond chemical compound, like astaxanthin has a double bond chemical compound, they can actually give their one bond electron or an oxygen to reduce that severity of we call free radical induced injuries. So here my proposal for this study will be, we are going to check three component. Natural strategy will be astaxanthin, which you've seen in salmons and algae of the cold water, and second will be omega-3 fatty acid. It has also shown to improve, especially incidence and severity. It decreases, especially when it comes to infections. Third one will be hydrothermal therapy, which will be could be very simple. We increase body temperature passively, externally or internally, up to 102. In healthy individuals, 102, 103. That's where macrophage and all the innate cells generally will be active. So it's generally training a primary innate immune system. and then you rapidly expose them to cold. It's called a evulsive therapy. It's called hydrothermal evulsive therapy, and it can be done into just by having a hot shower, sauna therapy, or anything. So there'll be a third group. Now, first group of the intervention will be astaxanthin alone. Second one will be, and of course, all of these drugs will be alone by itself. There'll be 3 groups, and in combinations. It has shown that astaxanthin combining with omega-3 fatty acid has shown a better bioavailabilities, and it has shown to decrease incidence and severity of viral infections in the past. So I think it could be a better approach to decrease hospitalization rate, because at this point, what we can do is propose a study to decrease incidence rate and boost up the primary immunity. I think it will be a better IND to submit to FDA. The only so far one clinical trial has been patented, and it was recently, but they are only targeting positive patients. We are going to target healthy or people who are at the high risk, other than healthcare workers, or will be the better population to target. Study duration we can keep up to 3 months, because they found that after 2 weeks of the astaxanthins and omega-3 fatty acid combined therapy, within 2 weeks they had a good signaling system, better efficacy in preventing, and even viral attachment also was decreased. They did the PCR, they did the screening for other viral tests in the past, and they showed that it has decreased incidence of those certain respiratory infection. Now, Spanish flu has in the past been a worse pandemic that we have seen, and they did hydrothermal therapy during that time, and they found especially during military, in the sanitary military, when they did a study of a hydrothermal therapy, that time they found decreased amount of incidence and severity rate among those military people, especially United States military people during that time, who were exposed to heat and were exposed to cold suddenly afterwards. It's called a hydrothermal evulsive therapy. So this entire study to do a single and cross check of the combined therapy just to see which one will be effective to prevent COVID-19.
Very good, Manu.
So this, if you say, I can include this, I can have, Kunal to, create this new IND, and I think it won't take, it's a least expensive. And if it comes to cost, finding population, people will not have an issue, because they all consider something we call GRAS, GRAS, which is generally relatively as safe compared to other therapy. And we can finish in a week if after creating. Anybody who is interested in this new IND, you can feel free to contact me, and we can start working with Dr. Michel's permission.
Excellent. So just let's set it up, that new Telegram group on that then, and take it from there. Excellent.
Okay. Thank you.
I've just reached out to Dr. Zamota, who is a resident at Larkin Community Hospital, and she's very interested in working with Dr. Madhusudan Patel's idea and on astaxanthins and hydrothermal therapy. So combining them with omega-3s. So she's on the meeting right now, well, actually.
Okay.
Sorry, can you repeat? Okay, sorry.
Hello, Dr. Zamota.
Yes.
So, as Dr. Patel was saying, we're looking forward to start a new study under the approval of Dr. Michel with astaxanthins and omega-3s, and we're glad that you're interested.
Sure, I'd be glad to find out more and work with you guys.
Perfect.
Sounds good. So I guess Manu, just make sure she's invited to the Telegram group, and then we'll develop a team for this. That's great.
Okay, fantastic.
Sir, do you still have one more, that last presentation, or?
The last presentation, so that is she's having difficulty with her speakers, but she sent me her draft, and I can do it.
Am I audible right now?
Oh, okay, we can hear her. Go ahead, Doug.
Okay, great.
Very good afternoon to everyone.
Yes.
I'm very sorry for the delay.
No problem.
So while reading about the covalescent therapy, I came across pathogen reduction therapy, which has been used to treat Ebola patients in Europe as well as in Africa. So basically what it does is before the transfusion of the blood or blood products, the products are treated with UV light so that the pathogens or any virus or bacteria which is there in the products is completely eliminated. So in routine testing, we cannot detect, like sometimes the infection is in its window period where it is not, you cannot detect it by routine testing, but it is still present in the plasma. So by this method, we can eliminate any such pathogen that is present in the product. And there are two kinds of therapies that are there. That is the intercept and the mirosol therapy. The intercept therapy is approved by the US FDA, but the mirosol is not. Mirosol is under the last stage of the human trial, I believe. It's waiting for its approval. And in the intercept therapy, they use a soralin derivative, which is added to the blood product, which is known as amatosalin. And then the UV light is added to the product and at around 300 nanometers of wavelength, and then given to the patient. So because coronavirus, we are still finding a lot of different like the natural history of this virus is not very well known. So maybe this can just add a protection layer to our covalescent plasma therapy.
Alex, does this interact with your UV trial?
I am not sure because I don't know enough information on it. One of the things I dug up was that they've shown that UVC at a fairly low level of exposure and time completely inactivates coronavirus in blood. That study was done pretty recently, but I have no knowledge. One of the ways that some people do ultraviolet therapy is that they mix in photosensitizers to make it have a stronger effect. That's most commonly done with cancer applications. But for the ease of simplicity, we were just going to go with ultraviolet blood irradiation and nothing else, since that's simpler and easier to do. You don't have to get approval for anything, and that should be sufficient.
Okay, I guess stay in touch because this might intersect somewhat with what you're doing.
Yeah.
Okay.
Okay. Okay, I'll get in touch with Alex and I'll just forward him my draft. If at all it intersects, then we can just combine the trial probably.
Yeah, or just have another arm, but at least even the uses what equipment you use for the UV and all that. He's already kind of advanced on that a little bit.
Okay, okay. I'll get in touch with Alex then.
Okay.
Thank you so much.
Thank you. Saurabh, any closing thoughts or?
So we are in the process to start the BCG1 trial pretty soon, within about 3 weeks now. So we're just gonna finish up the IRB consent letters all set, and then we are about done with 5 or 6 new INDs that we've described, and we have 2 or 3 new IND ideas. One being Dr. Patel's idea with the astaxanthins. So yeah, it's going great.
Perfect. Does anybody have any comments or questions?
Doc, I have a last comment. I'm doing the translation for the informed consent of BCG1, and I was wondering if there's a form in Spanish, because I mean, I can do the translation completely, but I don't want to like translate it in a different way that you have it already in a Spanish form.
Email it to me, and then I'll ask. I know Dr. Sanchez was sick, so I'm not sure if he's back to work already. But email it to me, and then I'll get it to the right person.
Okay, perfect. Thank you.
Thanks, everybody. So if no one else has questions, I've seen some activity on the chat. I mean, I think it's great that we keep coming up with new ideas. We should, and you know, you have my support. Let's keep this going, guys. Thank you so much for everything you're doing. I'm impressed with what you're able to do so far, and I just can't wait to see all the new ones coming up soon. I think Kruna just put in the Telegram and main FDA group. That's the team 11 one? Kruna, is that the one you meant? Okay, good.
Yeah, there are two groups. One is the Team 11 and one is the main FDI group. We can post it on either one.
Okay, yeah, go ahead and post. Okay, do
Yeah,
they?
I'll make the group and
Yeah, just go ahead and post it so everybody gets this cut and paste, and then because I've been hearing that some new people have been having difficulty joining the Telegram groups, so that would be great. We'll do that.
yes, we'll do that. Yeah.
Okay, perfect. You want to post it here, or you want to? I think I had posted it before. I have the one for the main Telegram groups. Going back on my, here we go. I have, this is the main Telegram group here. I'll post it to everyone. So that's the main Telegram group I just posted. And then I don't know if, Kruna, you can put the one for the main FDA group.
I'm
Sorry.
main link right now. I'll just add it to the chat. And
I
everyone.
wanted to suggest one another thing. It was found. It is yes, we know that the patent has been taken, and they are going. The company called will try clinical trials soon, but it is shown that astaxanthin is also at the higher dose works like a steroid. So if Madrol, in any Madrols or any steroid, that we are doing a study just to show the severity, and if they are still planning to, look for another control, astaxanthin also maybe you could check as a control, because that's not violating, any patent. You can still control and just see which one works better. That's just a suggestion, I think, for a Madrol study.
Excellent. Who's working on the Metro study? Sorry.
Karthik.
Okay.
Karthik and Karun.
So Karthik, maybe connect with Madhu on that. And
Okay, like we will do that.
Okay.
Because I think that way, we can do once, for example, hypothetically, if it generally get approved, we are planning study certain way, the result comes back, we can still do a cross-sectional studies. So the main aim is to, create studies certain way by cross-checking with other studies, and that will give us a better view. And in future, we can do a lot, it will help a lot of cross-sectional studies.
Definitely.
All right, so Karthik, I will update you, once, all the people, start working, I wouldn't mind, sharing the content. That way you can put as a control group in case, or you can send me a Madrol details, I will look over it and just to see, because the pathophysiology, the mechanism of actions are pretty much similar. They found even study that the efficacy of astaxanthin as steroid is equivalent when it comes to inhibiting interleukin-6 and TNF.
That's great. Definitely, I'll send you them.
And that goes to tocilizumab study too. That could be other study who wants to look for a control.
Okay.
All right, that'll be it from my side.
Thank you. Kuruna, are you done? Do you have anything else to put on the chat, or we're good?
Oh, we are good. We are good.
Okay, perfect. Saurabh, anything else?
That's all.
Okay,
That's all.
so the plan is we're going to do this monthly. So we're not going to do them every Monday so that you have enough time to work on your teams. Any issues or questions, obviously, I'm always checking the Telegram. So if you email me or message me directly, or you can also email me. I really want to thank you for all you're doing. I mean, I think this is a very exciting group to be in. And I just can't wait to get more and more FDA approvals for our projects. Thank you so much.
Thank you so much, Dr. Michelle.
Thank you, everybody. Have a great
Thank
great
you,
day.
Dr. Michelle. Have a
Bye.
good day.
Thank you.
Everybody.
Have a good day.
Thank you.
Thank you very much.
Thank you, Dr. Michelle.
Thank you.